Hi, I'm Lewis Quayle, data scientist, bioinformatician and maintainer of lewisdoesdata.com, which is my website where I blog data science tutorials and guided data analyses. Welcome back to this series on bulk RNA-Seq data analysis, where we're going step by step from raw sequencing reads to publication-ready figures. In the previous tutorial, we processed RNA-Seq data using the NF-core RNA-Seq pipeline and generated a gene-level counts matrix. So today we're going to be transitioning to local analysis using R. This session focuses on data import, exploratory analysis and quality control using DSeq2. And along the way we'll visualize the data, tackle data transformation and identify potential issues like outliers or batch effects. As always, the companion blog post for this tutorial includes links to all the tools, scripts and any further reading that I'd recommend. The first thing that you're going to need to do if you want to follow along, and if you didn't follow the first part of this series, is to make sure that you've set up your project directory and installed R and RStudio. For those of you starting here, instructions for how to do those things can be found in the first part of this series, and you'll also need to download the NF-core output archive from the linked companion blog post for this tutorial. Once you've done that, you need to transfer the rsum merge gene counts TSV file to the star rsum folder within the data processed subfolder. And then the metadata sheet needs to go in the data sample sheet subfolder. Next, you'll need to install the R packages listed in the script provided in the companion blog post, and there's also some code to verify those installations. Now, once we're all on the same page, we need to load the packages that we need, which will be DSeq2, ggRepel, ggtext, the here package for working with file paths relative to our project top level, and then the migrator and tidyverse packages. So I'm just going to run those lines. Then the next thing that I like to do is to set up a custom plotting theme, because we're going to be making some plots with ggplot2, and this prevents me having to write the theming code every time. And then finally we'll set that theme, that custom plot theme, as the default theme for ggplot2. The first thing that we're going to need to do in terms of progressing our analyses is to read or import the metadata and counts data that we obtained during the first tutorial into R. So when conducting any omics type experiment, such as transcriptomic profiling like bulk RNA-Seq, we refer to the descriptive data that outlines the biological and technical characteristics of the samples we've sequenced as the experimental metadata. Variables recorded in the experimental metadata will include things like cell line or patient IDs, the different control and experimental test groups to which the respective samples belong, the technical replicate number, and other variables like date of collection, to name a few. The importance of good metadata amongst providing sample labels and enabling correct parameterization of the models and contrasts needed for statistical testing later is that it should also record any potential confounding factors that we might need to address as part of our quality assessment. Because the metadata sheet's a CSV file and the counts matrix that we've made is a TSV file, we can use some of the functions from readr in order to import these data. So that's exactly what we're going to do when we run these two lines of code, and you can now see that we've got two objects within our environment. One is the counts data and one is the metadata. So if we want to have a quick preview of the metadata, we can do that interactively using the view function or clicking on the little grid up in the top right hand corner here. So once that opens in the viewer window, we can see that the metadata contains basic information about the samples that we'll need for our analyses. So we have things like the phenotypic conditions and the assay name assigned to the particular sample. And it also has several other variables earlier on, which are good for understanding the data set, but redundant in terms of analytical utility. So things like the organism and the cell type and the growth conditions in this case are relatively redundant pieces of information from an analytical perspective. The next thing that we're going to need to do is to clean up the metadata in order to get rid of some of those less useful variables and get those that we want to keep into a format that are ready for use later on. So what we're going to do here is we're going to take the metadata and we're going to pipe that into the janitor clean names function, which creates syntactically correct column names, and then we're going to select the two variables that we want to keep from that data frame. We're then going to use those, and more specifically we're going to use the assay name to create a lowercase sample ID by removing numbers using this regular expression here. And then converting them to lowercase. And then we're also going to create a group name so an experimental group to which the respective sample belongs. And then finally, we're going to create a replicate number. Now note that both the group and replicate are factors, so nominal categorical variables in R, and that becomes important later on. So if we run that and then interactively view the updated metadata that we've just created, we can see now that we have the original assay name and the phenotypic description, but now we've also created this lowercase sample ID to which we can use to link the samples to the respective count data, and we also have these group and replicate factors which enable us to know which experimental condition that the respective sample belongs to and which technical replicate that particular sample represents. Now that we've got the essential information from the metadata in the format that we need, we can move on to look at the counts data. So for the counts data, we have to transform it into a matrix compatible with DSeq2. So the first thing that we're going to do is to take our data frame and we're going to set the column names to the lowercase syntactically correct sample IDs that we created within the clean metadata. So you can see that's exactly what we've done here. The column names are now able to link each of the sets of counts for each gene to the respective sample detailed within the metadata. Then what we're going to do is we're going to remove this transcript IDs variable. So this is the variable which details the various different transcripts for which the counts have been made, and then those have been binned to the gene level. So if we run that, we can see that that column then is removed from the currently data frame or tibble that we have. And then the next thing that we're going to do is to take the gene IDs from a column and set those as the row names. So we can see now that the only variables that we have now are those that relate to the counts for each of the samples, and the gene IDs have been moved to the row names. And then finally, we coerce this data structure to the matrix type, and then round the values so that they're integer. So if we run all of that so that you can see that's the case, we can see now that we have a matrix of integer counts values, so we can assign that to the counts matrix object. And then just to be doubly sure, we can make sure that we coerce the storage mode for that data structure to integer. So if we preview that, we can see that we now have this matrix structure where the entries for each observation are integer level counts respective to each of the samples that we have in our counts matrix. Now that we have all of the necessary components, we can create what is called a DSeq dataset object. So a DSeq dataset object can be made using various different functions which come from the DSeq2 package. The one that we're going to use here is DSeq dataset from matrix, because we're creating this object from the matrix of counts that we've just seen. And then we're going to add the metadata, the cleaned metadata that we've created, and we're also going to specify the experimental design, which we'll come on to more later. So if we run that, we can now see that we have this object, DDS, which is of class DSeq dataset. The readout gives us the number of rows and the number of columns contained within that dataset. It also tells us which objects have been stored within the assay slot. It also tells us what the respective row names are. So in this case, the ensemble gene IDs that were set as our row names within the counts matrix. The column names are those matching sample IDs that the lowercase sample IDs that we created earlier on in both the metadata and the variable names of the counts matrix. and then the column data contains the metadata data frame that we created. There's a couple of useful accessor functions which you can use to retrieve the counts from the DSeq dataset, which are counts or assay. Counts is a wrapper around assay, which specifically will retrieve a counts matrix from a DSeq dataset, whereas assay is a more versatile lower level function which enables you to retrieve the assay slots from an S4 object. There's also an accessor function which allows us to retrieve the metadata from the DSeq dataset, and we can also access the individual metadata variables using the dollar syntax that those of you that have been using R for quite some time will be more than familiar with respect to data frames. And because we can access the individual variables using that syntax, it means we can also reassign individual variables using the same syntax. So in this case, if we'd forgotten to assign our groups within the DSeq dataset object, we could do that here. So if we quickly print out that variable, we can see that we've encoded the experimental groups as a factor, signified by the fact that we have levels and it's not just a character string. But if we'd forgotten to do that, if we'd forgotten to encode that particular variable as a factor, we could first coerce that variable to a factor, and then we could assign that to the group variable within the DDS DSeq dataset. So that's a quick overview of some of the ways in which you can access and manipulate the elements of a DSeq dataset. Now we're going to move on to visualize library size and count distribution. So the first step in our series of quality control checks is to determine how many reads we have for each sample in the dataset. The sum of each column in the counts matrix gives us the total number of sequence reads for that sample. So to get the total number of sequence reads for all sample, we can use the colsums function to return a named numeric vector. As we saw above, we can retrieve the counts matrix from the DSeq dataset object, and then if we run the colsums function over that, that gives us the total sum of all of the reads with respect to each of those samples. And because we have the variable names, what we've returned here is a named numeric vector. We can easily use functions from the tidyverse package dplyr to add these sample summaries to variables from the metadata to create a visualization of library sizes. You don't have to go to these lengths, a simple bar chart will do, but of course I like pretty plots. So what we're doing here is we're going to take the cleaned metadata and we're going to add a label by combining both the group and replicate variables. So we've added this variable here, which is a series of character strings. And then what we're going to do is we're going to left join onto this the name numeric vector that we've just created, but first we're going to turn that into, or coerce that into, a data frame itself, enabling us to do the join. So this in frame function with these additional arguments will convert that named numeric vector that we created into a small tibble or data frame, and then we can use the left join function to join together the augmented metadata that we've just seen with this, and we can join by sample IDs. So when we run those commands together, we can now see that we effectively have the metadata, the cleaned metadata that we created earlier with this additional label variable, as well as the respective library sizes, and now we're in a position to create our plot. So we're going to run this plotting code, which will give us a plot which shows us the sample library sizes for each of those samples in millions of reads. So we can see that there's a fair degree of variability across our samples, and this is something that we'll need to account for later on in our analysis. So similarly, it's possible to visualize the differences in distribution of counts data as, for example, a box plot. So we're going to take the counts data from the DSeq dataset object, transpose it, then convert this to a data frame. Having transposed it, the row names will be the sample IDs at that point, so we're going to convert that to a column, similar to how we moved the gene IDs to the row names earlier. And then what we're going to do is we're going to convert that into the long format. So what we'll have is we'll have the gene ID and the counts linked to the phenotypic data for every single one of the samples within our data set. And then finally we're going to add on a label variable for that. So you can see now that we've got the phenotypic data, and for every one of the gene IDs that relates to that phenotypic data, we have a respective count value for that particular gene ID. And the reason for doing this is because the ggplot function expects data passed in the long format. So what we're then going to do is we're going to map the samples, or the labels which denote the samples, onto the x-axis, and then we're going to plot the counts as millions of counts on the y-axis as a box plot. So if we run all of that code, we can see now that we've got these box plot summaries of the number of counts for each of the samples. What we can see is that visualizing the raw counts distribution reveals a small number of genes with very large counts, and these tend to dominate the distributions and will likely contribute very strongly towards differences in the libraries, which we saw in this plot here, because they're being quantified many times. And there are several biological and technical reasons why this phenomenon can occur. So those that are biological could be longer genes resulting in more fragments that can be sequenced. We can have higher constitutive expression, such as housekeeping genes, for example, and we can also have true differential expression between biological conditions. So it might be that these genes here are detected at much lower levels as a result of the experimental group to which the samples belong. The reasons could also be technical, so we have library preparation biases due to preferential PCR sequence amplification, or we could have differences in sequencing depth between the libraries themselves. Now, genes with high absolute counts can introduce a lot of variance, making it challenging to detect the true differences in expression for genes with lower counts, and this is why transformation techniques such as log transformation or variant stabilizing transformations are often applied to RNA-Seq data before downstream analysis. In DSeq2, the VST or RLog functions can be used to transform the data and compensate for the effect of different library sizes, and the effect of these transformations is to remove the dependence of the variance on the mean average and the high variance of the logarithm of count data when the mean is low. And we can see how transforming the data using, in this case, the VST function can be used to undertake a variant stabilizing transformation, and how this alters the difference in the distribution of the counts. So if we run this function here, that enables us to take a variant stabilizing transformation on the data contained within the DDS DSeq2 object, and we add this blind equals true argument here to ensure that the transformation is not influenced by the sample groupings or experimental design. And this is critical for maintaining an unbiased perspective during unsupervised quality control analyses. So once we've done that, we have this object which has the transformed counts data. We can then use the assay function that we mentioned earlier to access the transformed counts, and then we can undertake some transformation of the data. So we can transpose that matrix and convert it to a data frame. We can move the sample IDs over to a variable. We can left join that onto the metadata, just as we did in the previous plot that we created. We're going to take the same conversion to the long format, and then we're going to also add the label variable. So this is very similar to what we saw above. And then if we run that entire command to produce the plot, we can see in this case how undertaking that transformation of the data has altered the differences in the distributions of the counts. one of the primary reasons for having undertaken that is because possibly the most important quality control analysis that we can do is something called principal component analysis, or PCA, which is a fundamental technique in the analysis of bulk RNA-Seq data quality control and part of the exploratory data analysis process. So PCA transforms a set of possibly correlated variables into a set of linearly uncorrelated variables called principal components. And this transformation is accomplished in such a way that the first principal component will always account for the largest possible variance in the data, with each succeeding component or variable accounting for the highest variance possible under the constraint that it's orthogonal to the preceding components. So in the context of bulk RNA-Seq experiments, PCA is employed to reduce the dimensionality of the data while preserving as much variability as possible. A typical RNA-Seq dataset, such as the one that we're analysing here, comprises thousands of genes measured across several samples, resulting in a really highly dimensional data that's difficult to interpret directly. So what using PCA does is it allows us to simplify this complexity and visualize the data in a two-dimensional space, highlighting the main patterns in clustering between the samples and enabling identification of sources of variation as well as potential outliers. And PCA requires that we undertake this transformation of the data first, so that those genes with very high counts don't dominate the clustering pattern effectively. So DSeq2 has this convenient plot PCA function which allows us to pass in transformed data, specify a grouping variable of interest, and then the number of genes to use to compute the principal components, which is selected by the highest variance rows. And the default setting for this is 500 genes, the top 500 most variable genes. So if we run that command, we can see that we get this relatively rudimentary but perfectly serviceable PCA plot. Now there's also an argument that returns the underlying data and allows us to easily create a slightly more customized version. So you can see here that we've added this return data equals true argument, and we've passed in the variance stabilizing transformed data stored within the VSD object into that argument, and that returns the principal component, the first two principal components computed. So then we can pass that to the janitor clean names function in order to get some syntactically correct variable names. And then we can left join that onto the metadata and finally compose a data frame which we can use then to create a more customized version of the plot that the DSeq built-in function allows us to create. So the first thing that we can do is actually obtain the summarized percentage variance explained by the first two principal components from that PCA results object that we've just created. So that's stored in one of the percentage variance for those principal components is stored in one of the attribute slots, so we can pull that out using the ATTR function here. And as you can see, that gives us a decimal fraction of the amount of variance explained by each of those principal components, and then we simply multiply it by 100 to get it as a percentage rather than as a decimal fraction, and finally round it to 2 digits. So I've provided some relatively simplistic code to create a custom plot from these two objects that we've just created. So the PCA, the customized PCA results, and then the variance explained by each of the principal components we use to create an axis label here, pulling out the respective entry, and then we're going to combine those, concatenate those into a character string for the respective X and Y axes. So if we run that plotting code, we can see now that we've obtained a slightly more informative, slightly customized version of the default output created by the DSeq2 built-in function. So I go into a lot more detail about conducting PCA in the blog and how to create a nicer looking custom PCA plot. So the code for how to do that is linked within the blog post. Now, looking at either of these plots, you'll note that PCA plots generally display the percentage of variance explained by the respective principal component. So this is why we went to the trouble of creating that percentage variance vector. So we can see here that the first principal component accounts for approximately 64%, or the majority of variance in the data, and this actually appears to be related to our biological groupings, and we know that because the replicates for each of the groups, which are denoted here, are clustering together. So we have all 3 technical replicates for the IR group here, all 3 for the control group here, and all 3 for the TGF group here, and those are clustering along that that x-axis, that first principal component. So essentially what this is saying is that our experimental conditions are the largest source of variance in this data set, which is reassuring. So in essence, that's it for today. We've imported RNA-Seq data into R, we've undertaken data transformation, we've conducted exploratory analysis for quality assessment of the data prior to some downstream analysis, and in the next session we'll dive into now how to undertake differential expression analysis and adding gene level annotation. So I'll see you next time.